Hello, my students. Today's topic is transposable genetic elements. So, you will find this topic in your course code ZULH core course T12. So, today is the introductory session on transposable genetic elements, and at first, you have to know about what is gene. In case of definition of gene, we know that in very brief, it's a functional part of a DNA, and its position is very much pointed within a chromosomal locus. But in case of transposable genetic elements, we found it a little bit amazing kinds of genes here. Actually, transposable elements are unique mobile DNA segments in chromosome of both prokaryotes and eukaryotes, which are capable of moving from one location to another in the genome and can insert themselves at one or more new positions within the same or a different chromosome, leading to a change in the gene expression near it. That means it is a kind of gene which can jump. That's why it's also known as insertion sequences, or jumping genes, or nomadic genes, or controlling elements, or transpositions. Thus, it is found that transposable genetic elements are present in both prokaryotes and eukaryotes. So, in case of prokaryotes, we found three types of transposable genetic elements here. One is insertion sequence or IS elements, next is transpositions, and the third one is bacteriophage. So, in your syllabus, which cover only transposition, that is the one of the part of the prokaryotic transposable genetic elements. But before going to transpositions, you have to know about IS elements, which is a simplified a portion of transpositions. So I am going to the next slide, that is insertion sequences. This is a insertion sequence of transposable genetic elements, and I am giving you the slide or the figure of particularly IS1. We can see here that a picture of IS1 insertion or insertion sequence 1, and in this insertion sequence or IS1, the middle yellow portion is the gene, and the red boxes which we find in the left and right side, both in left and right side, that is written here is IR, which is the inverted repeats. So an IS element in prokaryotes is the simplest transposable element that contains only genes required for mobilizing the element and inserting it into a chromosome at a new location. IS elements are normal constituent of bacterial chromosomes and plasmids. Now we go to the properties of IS elements. In case of number per genome may be many in equally. There are a number of IS elements including IS1, IS2, IS10R. Each IS represent its 0 to 30 copies per genome, and each element with a characteristics length and the unique nucleotide sequence. Example, In case of IS1, there is which is 768 BP or base pair long, and it is present in 4 to 9 copies on equally chromosome. IS elements in prokaryotes range in size from 768 pp to more than 5000 pp and are normal cell constituents. Quantity of IS elements out of total genome we found altogether constitute about approximate 0.3% of the cell's genome. And the most interesting portion is the inverted repeats, as IS elements end with perfect or nearly perfect inverted repeats. In this picture, the IR represent the inverted repeats, and we can see that it is situated in left and right side, in both sides of the transposer's gene. The size of the inverted repeats in between 9 and 41 pp. This means that essentially the same sequence is found at each end of an IS, but in opposite orientation. Mind it, inverted, in case of inverted repeats, the unique feature of inverted repeat is essentially the same sequence is found at each end of an IS elements, but in opposite orientation. So it's about IS elements, and next we will in next session we will talk about transposon, the bacterial transposon, which is a kind of transposable genetic element found in bacteria. Previously I also informed you that in case of prokaryotes we found IS element transposon and other transposable genetic elements. So, and in your syllabus there is a transposon of bacteria only, but before going to the transposon, it was the introductory session. You have to know about IS or insertion sequence elements in a brief, that will help you to get to build up knowledge on transposon in next session. Thank you.